In this video, I'm going to go through the experiment carried out by Meselson and Stahl, which helped prove the semi-conservative replication of DNA. The reason this deserves a video on its own is that it is a popular experiment used in exam papers, and students often struggle to understand it. First of all, what do we mean by the term semi-conservative replication? Well, semi means part, so when the DNA is replicated, part of the original molecule is conserved in the new molecule. So the new DNA molecule is made up of one strand from the old DNA molecule or parental molecule, and one strand is a newly formed strand. There were actually 3 proposals of how DNA replicated: dispersive, conservative, and semi-conservative. Dispersive replication suggested that the 2 strands of the parental DNA molecule break apart at several points, resulting in several pieces of DNA. Each piece of parental DNA replicates, and then the pieces join back together randomly. This would result in 2 new DNA molecules, which are made up of patches of the original strand and patches of the new strand. Conservative replication suggests that 2 new DNA molecules are formed, but this time one molecule is made entirely of the original parental molecule and one molecule is made up of entirely new DNA strands. So now back to the experiment by Meselson and Stahl. In 1958, Meselson and Stahl wanted to prove the semi-conservative replication of DNA. We now know that in semi-conservative replication, the 2 strands of DNA separate by DNA helicase, breaking the hydrogen bonds between them. Each strand then acts as a template for the synthesis of a new DNA strand. In this experiment, Meselson and Stahl used nitrogen-15 and nitrogen-14 isotopes of nitrogen to show which nucleotides made up the strands in the DNA molecules. They decided to use these isotopes of nitrogen because they each have very distinct properties which enables them to be identified from one another. The nitrogen-15 isotope is heavier than nitrogen-14 because nitrogen-15 has a higher atomic mass than nitrogen-14. After centrifugation, DNA containing nitrogen-15 settles lower in the centrifuge tube than DNA containing nitrogen-14. Also, DNA with nitrogen-15 forms light-coloured bands in gel electrophoresis, as compared to DNA with nitrogen-14, which forms dark-coloured bands. Now let's go over the stages of the experiment as illustrated in this diagram. This diagram shows the results of each generation of E. coli bacteria that was cultured by the scientists. The flasks represent the E. coli culture, and the test tubes or centrifuge tubes represent the results after centrifugation. Then, in this column, you can see the composition of the DNA, and finally, the results from gel electrophoresis. In generation 0, Meselson and Stahl cultured E. coli in a medium containing nitrogen-15 only. This meant that all of the DNA in the E. coli was composed of nitrogen-15, so when the E. coli were extracted from the solution, broken up and centrifuged, all the DNA settled towards the bottom of the centrifuge tube as it was heavy. You can also see in the results from gel electrophoresis that there was just one single broad, light-coloured band. In generation 1, Meselson and Stahl added nitrogen-14 to the medium. When they centrifuged out their solution, they found that all the DNA produced settled in the centre of the centrifuge tube, showing it was of intermediate density. When run through gel electrophoresis, it formed a slightly darker band than generation 0, and it was further up the gel as the molecules were not as heavy. From this, they concluded that each DNA molecule had one strand with nitrogen-15 and one strand with nitrogen-14. This supported the semi-conservative replication theory of DNA. In generation 2, they kept adding more nitrogen-14 to the medium. When they centrifuged out their solution, they found that there were 2 layers of DNA of the same width, one near the top of the centrifuge tube and one near the middle of the tube. This indicated that half the DNA was still composed of one strand of nitrogen-15 DNA and one strand of nitrogen-14 DNA, and half the DNA molecules was composed of nitrogen-14 DNA. In the gel electrophoresis, 2 bands formed, one dark and one medium coloured, of similar widths. These results backed up those from the centrifugation. In generation 3, they grew the bacteria in a nitrogen-14 medium again. On centrifugation, they now ended up with a wide band of the least dense DNA at the top of the centrifuge tube and a thinner, medium density band in the middle. After gel electrophoresis, there was a wide, darker band near the end of the gel and a thinner one in the middle of a lighter colour, again backing up the centrifuge results. This is because the DNA with both nitrogen-14 strands will further form DNA molecules with only nitrogen-14 strands. The DNA molecule with one nitrogen-15 strand and one nitrogen-14 strand will form one DNA molecule with nitrogen-14 strands only, and one DNA molecule with one nitrogen-14 and one nitrogen-15 strand. This is shown in this diagram. And so this provided evidence for the theory of semi-conservative replication, which was further backed up by Cairns' autoradiography experiment in 1963 on radioactively labelled E. coli DNA, and Taylor's experiment in 1969 on radioactively labelled root tip DNA.